Caroline Uhler. So Caroline has a background in this very exciting areas of things like graphical models, statistical inference, these very powerful techniques that have come out of computer science that allow you to pull out patterns from complex data sets. And what she's going to talk about today is how to use these tools to think about a very core model in biology, which is looking at gene regulation and gene expression.
All the organizers for inviting me to speak here. I'm really excited to speak here in particular because it's a very different community than I'm usually in. And so I thought I should maybe start with giving you a little bit more background about myself. It's on. So let me start. Is it good? Yeah? Okay. So let me start by giving you just a little bit of background about myself. So I started off at the University of Zurich studying mathematics and biology. And then more and more I got into statistics because this gives me the framework to somehow combine the beauty of mathematical theory with applications to biology, which I find really interesting. Okay, and so what is this talk going to be about? So if you look at my publications, it will seem like I'm, a statistician, a mathematical statistician, and probably most of it has nothing or very little to do, or seems to have very little to do with any kinds of biological applications. However, all these mathematical problems actually, or most of these mathematical problems which I have been studying really came out of one particular biological question. And then, I had to take all these detours, and finally I can get back to this biological questions, which motivated me at the beginning. And so let me start this talk by just presenting this biological phenomenon, which I find really striking and has somehow caught my attention already very early on, and then tell you a little bit about the mathematical problems which arose out of this. Okay, so what is this problem? So here is this biological phenomenon which I find really striking. So we have so in each one of our cells, we have 46 chromosomes, 23 come from the mother and 23 from the father. And these chromosomes are the same in all cells in our body. And the genetic information is the same in all cells in our body. However, we have all these different tissues. Right? So we have, for example, brain cells and heart cells, and they look very different from each other, or liver cells, they look very different from each other. And also the products, just even the gene expression is very different in all these different cell types. So really just a DNA sequence is not able to explain any of this in some sense. Right? So there is much more behind it than just that. And so the ultimate question I would like to understand is, how is this possible? Okay, so what is it which really makes it possible to have all these different cell types, although the genetic information is the same? Okay, so then one way to represent. Okay, so I think at the heart of this question, what we need to understand is how do genes interact with each other. Right? That will tell us a little bit about what makes these different cell types really different from each other. And one way of representing, or the probably most common way of representing interaction between genes is given by gene regulatory networks, where the genes here are the nodes. Right? And a directed edge means that this gene up or down regulates this gene. And the edges can have weights. They are positive or negative weights. They tell us about the strength of this interaction, or the strength of so a positive edge would mean that this gene really upregulates this gene, or it can be a negative edge, meaning that it downregulates this gene. And so one goal here to get a little bit of intuition of how different are these different cell types is to try to learn tissue-specific gene regulatory networks. Okay, so nowadays we actually have tissue-specific gene expression data. So for different tissues, we do have gene expression data on the nodes. And so what we would like to do is learn these gene regulatory networks for the different tissues. That will tell us a little bit about the differences between them. But it does not tell us anything about the mechanisms behind it. Right? It will just give us a glimpse of what are the differences, but not what leads to these differences. So the first part will really be about learning how can we, in fact, try to learn these gene regulatory networks. And the second part of the talk is really about trying to understand the mechanisms which could lead to these different gene regulatory networks. And so here, one hypothesis has been that these different gene expression patterns are really caused by a different spatial arrangement of the genetic material in the nucleus. So the hypothesis is that if you have a different spatial arrangement, then different genes will come close together. And the hypothesis is that genes which are close together tend to interact more. And this then, of course, could lead to very different gene regulatory networks, just by somehow organizing the genetic material in a different way. Okay, and so the second part of my talk is going to be about models for the spatial organization of chromosomes. Okay, so this is a very broad problem, and I think all of us will use. If any of you would start studying this problem, everyone would use a different lens to look at this problem, right? And everyone will pick out the different kinds of subproblems which they want to study. And I'm going to use the lens of mathematical statistics and optimization and geometry to look at these kinds of problems and pick out the problems which I find interesting, and then show you a little bit about the results you can get by looking through this particular lens. But there are so many other problems around it where there will be need of many other different lenses to look at the same problems as well. Okay, so as I said, we'll have two parts in this talk. So the first part is about learning these tissue-specific gene regulatory networks, and the second part is about developing models for the organization of chromosomes, which could give us a little bit of an insight into what are the mechanisms which actually lead to these different tissue-specific gene regulatory networks. Okay, so let's start. So I said, okay, so the problem is to infer these gene regulatory networks just from observational data, so just from gene expression data. Of course, we can perform experiments, and this will give us some more insight into it. But you probably don't want to do a knockout experiment on each one of the genes to figure out your gene regulatory network. It just takes extremely long. So here I'll look at methods where we really just get observations on the nodes, and we would like to infer something about the underlying directed graph. Okay, and then we can ask ourselves, well, how can we actually make this process more efficient by actually adding in knowledge or prior knowledge which has been around from experiments. Okay, so if I want to learn directions, really what I'm asking for is I have observations on the nodes, and I would like to learn causal links, right? Directional links. And so this leads me to the theory of causal inference in statistics, and this has a very complicated history. So the framework was developed early on in the 1920s by Jerzy Neiman and Sewell Wright. They introduced a causal interpretation of structural equation models, which I'm going to show you. However, and they developed a lot of theory during that time. However, all of this got kind of nearly forgotten for about 50 years because statisticians were very, very skeptical about how one can and if one can infer causality just from correlations or just from observational data without doing experiments. But then finally in the 1970s, this whole theory came up again, in particular through really major contributions by 4 people. So Julia Pearl, Jamie Robbins, Don Rubin, and Peter Spertus. And in the last 10 to 20 years, this field has been very, very prolific in statistics. However, although there have been so many developments in this field, it is really hardly never used in practice. Okay, so the question is, well, why is that the case? Well, there can be many different reasons for it. Probably one of them that there has, and okay, hardly never used, I should say, it is used a lot in economics. That's probably the only area where causal inference is really used a lot, all these tools which have been developed in statistics. But why not in other sciences? Well, probably because there has not been the dialogue which should have happened from statisticians' side of going to domain experts and really explaining them what are the tools which have been developed. But probably also because statisticians have not been interested in developing methods which could directly be applied. I think many of them have just many of these methods really have some important drawbacks which make them like very difficult to apply to real problems. And we're going to see some of these problems now. But I really strongly believe that the interaction between genetics and causal inference could be extremely beneficial. Right? And why? I mean, why in particular with genetics? Well, because we know that genes are always causes and never effects. Right? So, I mean, we can actually use this in order to test our methods. Right? We can, and in particular, you know, it's pretty easy to do interventional experiments. Right? You might not want to do a knockout experiment on each one of your genes, but if I come up with a hypothesis of how the gene regulatory network works, then it's pretty easy for a geneticist to actually perform these knockout experiments and actually test the validity of these proposed gene regulatory networks. Okay, so I think there could be really a very nice interplay between statisticians of developing new theory and then actually also being able to test how well the theory is really doing in practice. And that's why I really like this area of genetics, because, you know, it's not like in many other areas where it's like unethical to actually perform these experiments and then test how well the, like for example, these things about smoking causes cancer. You don't want to be doing a randomized experiment and tell someone that they have to smoke for the next 20 years and see how it's going to do. Right here, you can actually perform these knockout experiments pretty easily. Okay, so let me give you a little bit of background on causal inference. And let's maybe start with, I mean, just because I said that probably the problem of why it has never been applied is because many of the methods actually don't have the qualities which you might need or really desire for actually applying them to genetic problems. So let me just start. So I made a list of 4 points which I think are really important, which a causal inference algorithm has to satisfy in order for it to be applicable to genetics. And maybe you will have other ideas, and you know, it would be good to make this list longer just so that I know what I should be looking for. So first of all, I think all the algorithms, well, should be applicable to very large data set. So I need to have, you know, I have like many genes. So the algorithms which will be used need to be algorithms which are actually applicable to graphs with about thousands of nodes, not just tens of nodes. Another problem with pretty much all causal inference algorithms are that they assume directed acyclic graphs. So what is that? So they assume that the graph has no feedback loops. Okay, that it cannot have any directed cycles. So graphs like this are not allowed. Here I have a directed cycle. Okay, I have a feedback loop. These things are not allowed in pretty much all of the causal inference algorithms which are around today. What is allowed is something like this. Okay, I can have cycles in the underlying undirected graph, but here I don't have a directed cycle, right? I have like a point here where everything comes together. So it's not a directed cycle. So these are allowed. These things are not allowed. Okay? So most algorithms assume a directed acyclic graph, so no directed cycles. However, I think feedback loops are very important in any kinds of biological networks. So it will be important to develop methods which somehow do not make use of this assumption. Another point is that, well, causal inference in itself is already so difficult that you want to be able to increase power of all these methods by actually making use of prior knowledge. I mean, people have performed experiments and know for some genes which ones are upstream from others, and so we should be able to actually include this kind of prior knowledge in the method itself, or make use of it, at least when we have it. And another point which is important for me, I think it is, I really don't want to be just using heuristics where you have no idea when the algorithm is going to output the correct graph. So I think it is important that we have algorithms where we know that under certain assumptions it is going to output the correct graph. And then you can test if these assumptions are actually satisfied or not. So you need to understand when does an algorithm, because everyone can come up with an algorithm. I mean, anyone can come up with a heuristic, but it is kind of dangerous of just proposing heuristics where you have no idea when they're actually going to work and when not. And so let me just tell you a little bit about the two main approaches which have been around in this area. So the first approach are Bayesian approaches. So what do they do? So you have, so both of them actually work on DAGs, okay? So not with directed cycles, so directed acyclic graphs. And so what does the Bayesian approach do? So you have the space of all DAGs, which is huge, okay? And now you wander around the space using heuristics. So you, for example, start with an empty graph, and then you add in the edge, which increases, for example, the likelihood or the penalized likelihood the most. And you go on, okay? You choose the next edge, sometimes change the directions, and so on. And you just wander around the space like that until you maximize some kind of score function, any kind of penalized likelihood, BIC, AIC, anything like that. Okay, any score function will work. Well, okay, so what is the problem? Usually does not come with any provable guarantees. However, it is very easy to include prior knowledge. I mean, this is really a very important point for these Bayesian approaches, that they can, in fact, incorporate prior knowledge. Again, they do not usually include feedback loops. And also because they're wandering around this huge search space, okay, you can apply it to graphs with thousands of nodes, but usually you have a problem that you're actually not going to explore the whole search space just because it is too big. Okay, so that's one kind of class of algorithms for causal inference. The other class of algorithms are constraint-based algorithms. Here, the main algorithm, and probably also the most widely used algorithm in all of causal inferences, is called the PC algorithm. I'm going to explain what it does. Here, this algorithm is, so it comes with provable guarantees. So it is known to be consistent. So it is known to give you the correct graph if you have data from a certain DAG under one particular assumption, which is called the strong faithfulness condition, and I will explain what it is. So it does come with provable guarantees. It cannot include prior knowledge. I'm going to explain to you why it cannot include prior knowledge. Again, it does not allow for feedback loops, but it is really applicable to very, very, very large data sets. So this has been used in high-dimensional statistics with 100,000 nodes. So, but here we really have to, okay, so here we have provable guarantees, but we should probably ask ourselves, how strong is an assumption? Okay, it works under this assumption, but when is this assumption actually satisfied, and can we hope that it is satisfied for these gene expression data sets which we would like to analyze? Okay, so these are the two main approaches. I will just describe a little bit about what this PC algorithm does, and and also describe about why is it not true that from correlations you cannot get any kind of causal inference? Okay, I mean, yes, one has to be careful, but I think in particular in biology, I mean, people are very, very skeptical about these things. You cannot get causal links from just correlations, but you can get some directional information. Okay, and I'm going to show you how you can actually get some directional information. And then also explain how the PC algorithm works. Okay, so let me show you how you can get some directional information just by doing it through one particular example. Okay, so we have observational data. We would like to determine the causal structure. So all we have is correlations, partial correlations. Okay, so, or conditional independence relations. So what do I mean by, so first of all, First of all, I have to just tell you what all these signs mean. So I have here, this means independent. Okay, so knowledge of X does not provide any knowledge of Y, and vice versa. This is a conditional independence. So assuming I know this variable, then knowledge of X does not provide me any further information on Y other than what I already know through Z. Okay, so this is just how I represent conditional independences. And so let's look at a particular example. Okay, so here I have a DAG. You see I don't have any directed cycles. And this is just a family tree. Okay, so I have mom and dad. They have two children, Linda and Henry. Linda also has a child called Carl. Oops. And let's think of the variables as just genetic information. Okay, they're genomes. Okay, so and let's start looking at two variables. For example, Linda and Henry. So they're sister and brother, right? So they're related. So definitely their genomes are not independent, right? Knowing Linda's genome does provide me information on Henry's genome and vice versa. However, once you condition on mom and dad, then they become independent, right? Because knowing mom's and dad's genomes, then knowing Linda's genome does not provide me any further information on Henry's genome, other than what I already knew through mom and dad. Okay, so they're conditionally independent. Now we can look at other examples. So for example, let's take the grandchild and grandma. Situation is the same, right? They're related, so they're not independent. Once I condition on the nodes in between, then they become independent, right? I cannot get any further information from Carl's genome on mom's genome if I already know Linda's genome. Okay, so now let's look at a last example. I'll take mom and dad. Okay. Well, usually they are unrelated. So then their genomes are independent. Okay. However, and now something interesting happens. So before it was always like when I'm conditioning on nodes in between, they become independent or they remain independent. Okay. So now I'm conditioning on nodes in between, right? You see these paths here. I can condition on any one of them. Let's take Henry. When I condition on Henry, they actually become dependent. Right? Why is this? So assuming I know Henry's genome, then knowing mom's genome does actually tell me something about dad's genome, right? Because everything which Henry has, but mom doesn't have, has to come from dad. Okay? So suddenly by conditioning, you're actually introducing dependence. And this happens always when you condition on any node which is a descendant of two nodes. Okay? So even if I would condition on this guy or I would condition on this guy, they are becoming dependent. So whenever I condition on a descendant of nodes, they actually become dependent. Okay? So this shows you that, just from these kind of conditional independence relations, I can actually say something about directions. Okay? I can say something about causal lengths. Just from observational data, right? All I'm observing is, I'm not doing any experiments. All I'm observing is just the nodes, data on the nodes. And this does give me some information about directional links. What you cannot do is if I just have two nodes and I want to know if the edge goes this way or that way. This I'm not going to be able to tell, right? But you can tell something about these paths of length too. There you can say something about the directions of these edges. Okay. So that was the first thing. So now, what does the PC algorithm do? It has to go backwards, right? It doesn't know this. All it gets to see is like independence relations or conditional dependence relations like this. And from this information, it wants to learn the graph. Okay. So how does it do it? Well, it just starts with a completely connected, undirected graph. And it just goes through all these conditional independence relations. And whenever it finds an independence relation, it just removes that edge. Okay, so let's...
There are some genes you can't, not right for imprinted genes.
This is not right for imprinted genes. Oh, because of this, you mean?
Well, because if there's an imprinted gene with the mother, then knowing about Henry doesn't tell you anything about it.
Yeah. Okay, so this is...
There's some caveats.
Yeah, sure. This is just to tell you a little bit about the idea of, that you can actually do things like this. That there is a difference on, when you condition on descendants, that you can say something about the structure of the graph. Okay. Okay. So let's again say. So how does the algorithm work? So I start with a completely connected, undirected graph. Whenever I find any conditional independence relation, like for example this, I'm just going to remove that edge. Okay. When I find that, I'm going to remove this edge, and so on. Whenever you find a conditional independence relation, you just remove the edge. Now, of course, I didn't list all the conditional dependencies, but this is how it would work. You just always remove an edge. And so that gives you an undirected graph. It actually gives you the skeleton of this graph. And then you use exactly the arguments which I was now going through to orient the edges. Okay, so you look at guys which are somehow 2 steps apart, and you use this kind of information in order to orient the edges. Okay, so that's how the PC algorithm works. Now, well, this means that the PC algorithm requires one main assumption, right? Because it's always going to remove an edge whenever it finds a conditional independence relation. So missing edges have to correspond to conditional independence relations. Otherwise, it's making a mistake. And that's exactly what the faithfulness condition says, that missing edges have to correspond to conditional independence relations. Okay, Now, it might not sound like a very strong assumption, but when is this assumption not satisfied? So let's just make an example. So let's say I have some medicine, which helps for, I don't know, some lung functioning or something. Okay. So it has a positive effect on your lung functioning. But let's say it's also a super strong medication, and it has like a negative effect on your immune system. Okay. Well, and a well-functioning immune system, of course, has a positive effect on your lung functioning. A badly working one, probably a negative effect on your lung functioning. So now, what does the faithfulness assumption say? Well, it says that causal paths cannot cancel out. Okay? It says that I am not allowed to find that medicine and lung functioning are independent from each other, although there is an edge between them. So it says that this path and this path cannot cancel out. Now, this is kind of an important or a pretty strong assumption, I would think, for gene regulatory networks, where you probably have things like this, where one gene strongly downregulates a gene, but it upregulates another gene in between, which also upregulates this gene. And through these different paths, you actually have cancellation of these causal links. But this PC algorithm will make a big mistake. So let's say you have these cancellations. You will learn that medicine is independent of lung functioning. What it's going to do is it's going to remove this edge. Okay? And it learns that they are independent. So we're exactly in this case here. It learns that they're independent, and they are dependent when I condition. So what it's going to do is actually, it's even going to learn the directions wrong. Okay? So, this is probably the worst mistake you can do in causal inference, right? You might not care that much about actually not figuring out a link, if it is like maybe a very weak causal link. But you definitely care a lot about making mistakes like this, where you're getting to get the causal direction in the wrong direction. Okay, so the problem with this PC algorithm is really that it somehow first learns an undirected graph, although what you really want to get right is the directions. But when you make a mistake in learning the undirected graph, it will lead to very bad mistakes upstream. Okay? Or it can lead, sorry.
You also add links or just remove them?
No, you usually only remove. So one thing is actually always satisfied. So if I have a missing edge, this will always give me a conditional independence relation. That holds always. The problem is the other direction. When I have a conditional independence relation, the faithfulness assumption is really this. When I have a conditional independence relation, it means that I have a missing edge. Okay, so I might have additional conditional independence relations which just lead to me removing too many edges. So that's what happens in the PC algorithm. But then, okay, I would be okay with that, but not with it actually learning directions wrong.
So is this all in a framework where the data that you're given independent correlated, or do you actually get numbers for how strong the correlation is?
Oh, you get, yeah, okay, so we'll exactly get to that. For now, I have perfect data.
An exact cancellation is sort of unlikely, but...
Very good, I'm exactly going to get to that. Okay. For now, I have perfect data, and the problem occurs when I have perfect cancellation. But of course, we'll talk about, data is never perfect, and what happens then. Okay, the faithfulness assumption is in the perfect setting, or I have exact cancellation.
Actually, those are two questions, right? One is about errors in the data, and the other is about whether you're being told discretely correlated, uncorrelated, versus being given a number.
But it's actually the same question. So when you have perfect data, then you know if you're independent or not.
Okay.
And then when you have noise, you will have to include a cutoff value. Right? Okay. Okay, so this is the intuition behind the faithfulness assumption. And so I think it is really important. Yes, there is an algorithm with provable guarantees, but we should ask ourselves how strong of an assumption is this assumption. Okay, and I would like to just give you a little bit of intuition for what this assumption means. And so until now, I didn't have a model behind this, or a formal model at all. So let's introduce a formal model. And the model I'm going to use is these linear structural equation models, just with Gaussian noise. Okay? So I will have like a very complicated slide, so let me just explain it. So let's call these guys x1, this guy x2. Let's turn it around again. x3, x2. Okay, so what do I mean by it? So think of gene expression. Maybe take a logarithmic transformation on your gene expression. then what is this model? So here I don't have any incoming edges, right? So this is just, x1 is just some Gaussian noise, okay, because we don't have any incoming edges. Then x2 has one incoming edge, and all the edges will have some edge weights. Let's call this A12. So then x2 is, the causal effect of x1 on x2. So this is the strength. It can be positive or negative. And then we have some Gaussian noise. Okay. And then we have x3 there. It has two incoming edges. They have some causal strength. Okay. So x3 would be, the causal strength of x1. So A13 times x1 plus A23 times x2 plus some Gaussian noise, okay, where epsilon just follows a normal distribution with some diagonal covariance matrix. Okay, so this is all independent Gaussian noise. It has some different variances. But this is going to be a very simple model, which I'm going to use in order to quantify how strong this faithfulness assumption is. It's probably a model which pretty much makes sense for gene expression data. You have continuous data. After logarithmic transformation, you'll have positive and negative values, and then maybe makes sense that we just have Gaussian noise here. And if I write this out in statistical terms, okay, so this is already in statistical terms, but okay, so here is the formal definition. So I have a graph, a directed acyclic graph. I have p vertices. I have a set of edges, directed edges. Now, because I always assume that there are no directed cycles, I can always order my vertices as I did it here, so that smaller vertices can only point to larger vertices. Okay, this is always possible because I don't have any directed cycles. Then I have edge weights. These are the causal effects, which I also had here. On each one of my edges, I have edge weights. And then I have samples. So I get to see some samples here. So now let's figure out what is the distribution on the axis. Well, I could take everything to one side, right? Only have epsilons on this side, where I know the distribution. So if I do that, so here I have x2 minus a12 times x2, x3 minus all of these guys, and you'll see that I get this kind of equation, right? So I have the identity of x, I have these guys, and then I'm taking all these minuses of all these other x's, which have some weights in front, where this matrix A here just consists of all these entries, right? A12, A13, A23 in the right position. So this is just my matrix A here. Okay, so you'll see that we can actually read off the distribution of my x's, right? So this is a normal distribution, and the distribution of x is still a normal distribution. The covariance matrix changed by this amount. So what we find is that, so I have samples from a multivariate normal distribution where the inverse of the covariance matrix has a very special form. Namely, it is this I minus A times some diagonal matrix times I minus A transposed, where this matrix A here is just the weighted adjacency matrix of my DAG. Okay? So that's all you need to know. So it comes from a multivariate normal distribution. This covariance matrix has a special form. And what I want to do is I'm given samples, and I want to learn the DAG. Right? So what I want to learn is I want to learn this weighted adjacency matrix. I want to know where are there zeros. Right? Because zeros in here mean that this edge here is missing. So I want to learn the correct ordering, and I want to learn this upper triangular matrix A. Now, maybe you have already heard that every positive definite matrix has a unique factorization of this form here, where it is upper triangular times diagonal times upper triangular transpose. Sorry, it's called the Cholesky decomposition. So every positive definite matrix actually has a unique such decomposition. So the problem really is to figure out what is the correct ordering and where are the right zeros in this Cholesky decomposition. Okay, and just maybe one more thing. So I told you that we're going to learn a DAG from conditional independence relations. So what are conditional independence relations in the Gaussian setting? In the Gaussian setting, they just correspond to the vanishing of partial correlations. And actually, partial correlations have a very nice algebraic representation. They're just the vanishing of certain minors in the inverse covariance matrix. And maybe, I mean, all it is like you take your inverse covariance matrix, you remove some edges and some columns and some rows, and then that subdeterminant is vanishing. And there is really a one-to-one correspondence between the vanishing of partial correlations and this very nice algebraic term, which just says you take a subdeterminant and you set it equal to 0. Okay, this was just a little bit of math background, but let me now give you some, I don't know, if you've never seen this, and maybe this is too fast, but still you will be able to understand, let me just tell you, oh yeah, sorry.
So this is correlation in time, right?
Here I don't have actually any time, right? I just
get... What does it mean, correlation? Relation, no, I understand, but from the data, how would you infer this?
Oh, so...
many samples, and
yeah.
you're going to use multiple samples to figure out whether they are correlated or not.
Yes, exactly. So...
But that's not...
Well, this is exactly what I showed you on this slide, right? So, okay, so what I'm going to do is, so I have n samples from this model, okay? So I have n of these samples. First of all, I can compute the sample covariance matrix. Okay, so I get a big matrix of size p by p. Just that consists of my correlations. Okay.
That's not correlation in time.
No, just correlations.
Correlations in average, so that's kind of a steady state correlation.
Yes, exactly.
Across many cells.
Mm-hmm. Correct. Yeah. Okay. So these are my sample correlations, is what I get in this matrix. But I can also get my sample partial correlations. Okay. So these are these kinds of guys, the correlations corresponding to when I'm conditioning on a subset. They just correspond to. So I take my sample covariance matrix, and I just, remove some of the edges, I take a subdeterminant. Those are going to be my partial correlations. Now my partial correlations, whenever they're close to 0, I can say that I have an independent statement, and then I do exactly what I was showing you before. Is that okay? Okay, so everything is based on the sample covariance matrix. Okay, and so now let's get some intuition for what this faithfulness assumption means. Okay? So the faithfulness assumption means that whenever I have an edge, I cannot have a conditional independence relation. So if this is my true graph, then I'm not allowed to have any conditional independence relation. Okay? Because otherwise, the algorithm is going to make a mistake. It's going to remove an edge when there is an edge. So what are all possible mistakes the algorithm could do? So what are all possible conditional independence relations on 3 nodes? There are just 6 of them. Okay, there are 6 of them. I can just list them. And I can write out the algebraic expression, what it means to have this conditional independence. It's just a minor which vanishes, right? Just a subdeterminant which vanishes. Now, I want to be able to draw the space of all unfaithful distributions. So I'm going to make just a little bit of an easier assumption. I'm just going to set the variances of all the noise to be equal to 1. Okay, so I have independent IID noise where all of the variances are equal to 1. I just want to cut down my number of parameters so that now I'm left with only 3 parameters, 1 for each one of the edges. So I can then write down what is my inverse covariance matrix, right? It's just I minus A times I minus A transpose. I just multiply it out. That's all I did. I just multiply it out, and this is what I get. Okay? Now, as I said, every conditional independence relation just corresponds to a subdeterminant. Let's maybe not go through the math. But what does this show you? So the algorithm is going to make a mistake whenever any of these conditional independence relations holds. So whenever any of these polynomial equations are satisfied. Okay? So what are these so polynomial equations correspond to hypersurfaces. Right? I did it in 3D because I want to draw it. Okay? I only have 3 parameters, so I can actually draw it. So what you see here, so we have 3 of these very simple polynomial equations, which just correspond to the parameter hyperplanes. Right? And then I have 3 more twiggly guys, and I can draw them, right? I'm in 3D. And so here is a picture of it. So you see here I have in pink, I have my 3 coordinate hyperplanes. And then I have in purple, red, and green, I have my more twiggly guys. Okay, so on 3 nodes, this is the space of all unfaithful distributions. Okay, in the Gaussian setting. So whenever a distribution lies exactly for now on one of these hypersurfaces, then the algorithm is going to make a mistake. It's going to remove an edge, and in general, that will lead to a wrong DAG. And now I get to your point, okay? So, well, as you were saying, it's not usually the case that something will exactly cancel out, right? So why am I even saying that this is a problem? Because, the probability that a sample distribution actually lies on here is 0, right? Hypersurfaces have measure 0. So they will never, ever get to lie on these hypersurfaces. So why am I even saying that there is a problem? Okay, so data is not exact, right? So even if there should be a conditional independence, the sample conditional or partial correlation is never going to be exactly 0. So when you work with data, you will have to define some cutoff value. You will have to say that, well, every partial correlation which is smaller than some cutoff value, which I will call lambda, is just going to be set to 0. Otherwise, you'll never get a conditional independence relation. Now, this cutoff will, of course, depend on how many samples you have. So if you have an infinite amount of samples, then you can choose lambda to be equal to 0. If not, then you have to, it scales as 1 over the square root of the number of observations.
Yeah. The coordinate, the planes themselves correspond to one of these things is 0. So I might have missed something, but if one of those is 0, then it should remove the edge. Should it not? But you're saying that's a mistake.
Okay, so one of them being equal to 0. But, this corresponds to it being equal to 0, although it should not be equal to 0.
I see.
Okay, so when you have samples, you will be close to it being equal to 0, but the edge should actually be there. That's why it's a mistake. Okay, this is the correct model, but what I'm going to infer is that the edge is not there. Yeah, very good question. Okay, so now, as I said, so I have to include this cutoff value, where I set everything which is smaller than this cutoff value, I'll set it to 0. Okay, well now, now what this strong faithfulness assumption is just a data version of the faithfulness assumption. Okay, it says that, well, what the algorithm is going to do is, whenever you infer that something is 0, it's going to remove the edge. So strong faithfulness means that you're not allowed to be close to any of these hypersurfaces. Because when you're close, I'm going to infer that the partial correlation is 0, and I'm just going to remove the edge. Okay? And so now, of course, well, this does not have measure 0 anymore. Okay, there will be many many points which are close to these hypersurfaces. So now the question is how many of them are actually left. Okay, once I fatten up all these hypersurfaces, what is left? Okay, what are all these distributions which are actually left? Okay, so the first thing is, well, how do fattened up hypersurfaces actually look like? And that was probably my first surprise. I mean, I just didn't think about it. But once you start plotting them, let's do that, okay? So I'm going to take a polynomial, very simple polynomials. Okay, that's what I get from my subdeterminants. And I'm going to plot all the points such that, let's do it. I'm just going to do it in 2D. So I'm going to plot all the points such that a polynomial is smaller or equal to lambda. Okay. And so probably most of you will think that the hypersurfaces are just going to be fattened up everywhere in the same way. I mean, that's at least what I expected. It's not the case, okay? So I'm taking very, very simple polynomials. So f of x, y is equal to x. I'm looking at all points which are close to this hypersurface. Here I'm taking another one, x times y, so this is the hypersurface. And I'm looking at all points which are close to it. Okay, another one, another one. So you see that there are special points which are fattened up much more than others. These special points are singularities of the hypersurfaces. So what we are interested in is figuring out, once you remove these fattened up hypersurfaces, then out here is good area, right? Where we're not close to these hypersurfaces, that's good area. That's what we want to quantify. How much is left? So we can first ask ourselves, well, let's do it asymptotically, okay? So let's let the number of observations go into infinity, meaning the fattening goes to 0, and figure out what is then the volume which is left. Okay, so if we let the fattening go to 0, then you see that the whole volume, or the behavior of the volume, is just given by these most fattened up points, which are the singularities of the hypersurface. And in fact, there is a very nice theorem by Watanabe. So this actually uses a very hard algebraic geometry. So it says the following. So it says in the asymptotic setting, okay? So if I let lambda go to 0, which is the same as the number of observations go to infinity, then this volume is actually asymptotically given in this form here. So it is some constant times the fattening, lambda to some power, times minus log lambda to some other power, where these powers here are really characteristics of the singularities. So algebraic characteristics of the singularities. They tell you how bad a singularity is. Okay, so you see already here, somehow this volume and this volume, they become always fatter and fatter. This is actually because the singularity is worse in some way, which is characterized by something called the real log canonical threshold. Okay, so algebraic geometry will actually give you exactly what happens, or how large this volume is when n goes to infinity. However, in our applications, we probably don't care about this, right? So we're in this gene expression framework where, compared to the number of nodes, number of observations will not go to infinity. You usually have much fewer observations than the number of nodes. And so this is probably not the kind of framework we want to be studying. This is important for many other applications. Okay, but let's see what happens if we have finite number of observations. So lambda is pretty large in that setting. Okay, we have very few observations, so lambda is definitely not going to go to 0. So it's scaling as 1 over the square root of the number of observations. Okay, just to get a little bit of an intuition for how small this volume is, which is still left over, let me just show you some simulation results first. Okay, then we can actually get some theoretical results. Okay, so what are these simulation results? So I start with very small graphs. So 3-node DAGs, 5-node DAGs, 10-node DAGs. On this axis, I have the density of the graph. So 0 means no neighbors. 2 in this case means full graph. Okay, so it always goes from very sparse graph to very dense graph, same here. And then here, I have the proportion of unfaithful distributions. So the distributions which are close to the hypersurface. And I do that for different values of lambda, which is the same as saying different values of the number of observations. Okay, so in blue, it corresponds to 100 observations, in red to 10,000 observations, and in green to 1 million observations. Okay, so what do we see? So first of all, the more observations, the less observations you have, of course, the more you have to thicken up, so the more this volume increases. Okay, so that makes sense. But then there are some problems here, right? So I think this is pretty small graphs, already pretty sparse graphs. I mean, already just 2 expected neighbors, a lot of observations, like already 1 million, or here, let's say 10,000 observations. You have hardly no more distributions which actually satisfy this assumption. Okay, so out here, this is in fact a 0. Okay, so this means that there is no more space left. So this means that every sample will actually give you the wrong DAG.
If the distribution doesn't satisfy the faithfulness assumption, you're saying it's really bad because then the errors could propagate to the entire inference. It's not even that just a fraction of these things would be.
I mean, I can make some cases, of course, if you just have one edge, and
sure.
It's going to be different.
But the reason you're looking at, the entire distribution has to be faithful is because even if it violates in one place, it could be arbitrarily bad. Okay.
Every single, so this is really, I mean, a necessary assumption. So every mistake is going to have this effect of like, you remove an edge and then you're going to include.
And I could ask this later, but is this lambda, you put the lambda in and then you connected it to finite observation size, but you haven't really made that very formal. You're just taking lambda as one.
No, this is known, like lambda has to be of this order in order to get the partial correlations right. Okay? In order to get consistent estimates of the partial correlations. So that has already been known long before. Yeah.
All the distributions that remain, all the networks that remain after you've removed the ones that are not appropriate, they're all effectively the same, right? They all explain the data.
Okay, so here it's not networks which I'm removing. Here it's like for every network you have a certain
whatever
set
connections
of distributions.
whatever distributions are left are believed to be representative of the data, right? But
yeah,
there's
so
only one single true distribution. How will you ever find that?
Okay, so here the problem is, so I start with a DAG. Okay, so in this setting I know what the true DAG is. So corresponding to this true DAG, there are many distributions, right? I can choose my parameters to be anything. Right. So there are many true distributions.
Okay, so you're just making bounds on the parameters. That's really what.
Yes.
What
Okay.
is the
Yes, you
goal
can
of
see like
this?
that.
Well, the goal of this is to find which distributions are consistent with the data. Is that all it is?
Well, for which distributions am I actually going to learn the correct DAG? Okay, so all of these distributions, I can choose these to be whatever I want them to be. They're going to be consistent with the DAG, right?
I see.
But my question is like, when am I actually, so this is a particular algorithm, right? So this algorithm needs faithfulness. So this algorithm says that, you know, the distribution, the sample distribution really has to be bounded away from many of the true distributions are not going to be okay because the algorithm is going to learn the wrong DAG. Okay? This is an algorithm thing. I mean, it's just for this algorithm that I'm showing that, you know, this is the most widely used algorithm in causal inference, and it is usually going to output the wrong graph. Okay. So maybe you can ask yourself, well, why wasn't this known? Why did nobody test this? I mean, if you come up with an algorithm, you should probably test your hypothesis. But, you know, I mean, this causal inference has a very complicated history that it was always between philosophy and statistics. And the people who actually developed this algorithm are all philosophers. And I mean, it makes sense for them that they don't care about when this assumption holds or not. I mean, it was like a pretty big development to actually come up with this algorithm. Right? I mean, these are super smart people, and this is a very nice algorithm. And they ask themselves very different questions. They never thought about actually applying these kinds of algorithms. So that's probably why, you know, these things can actually happen. Okay, so let me maybe not say anything. So we can actually then get, you know, theoretical lower bounds. I mean, I can prove to you that this algorithm is usually going to output the wrong graph, and, you know, I can tell you that, you know, this happens, for example. So when the numbers, just for trees, is the simplest situation, when the number of nodes increases, then this volume of unfaithful distributions actually converges to one exponentially in the number of nodes. If you have more complicated graphs where we have a lot of loops, then this actually increases to one doubly exponential in the number of nodes. So there is like just no way of this actually being able to be used. I mean, you can of course run it in the high dimensional setting, but you're just always going to get garbage. Okay. So this really means severe limitations for the PC algorithm in the high dimensional setting. And so, I mean, so now we have two kinds of different approaches for doing causal inference. One has no provable guarantees, one has provable guarantees, but under assumptions which just never hold. So then the question is, well, but maybe this is the best we can do, right? I mean, it's easier for me to tell that this doesn't work, but maybe this is the best. Maybe these are really the limitations of any causal inference algorithm, that you need to satisfy these faithfulness assumptions. So we should ask ourselves, well, what are the limitations? What are really the basic, the minimal information theoretically, the minimal assumptions I need in order to learn a DAG from just observational data? So I'll show you the algorithm which actually does this. And I'll show you that it is, so you can come up with an algorithm which has strictly weaker conditions than faithfulness. And so these minimal assumptions are actually strictly weaker than faithfulness. Okay? But we'll have to pay a price. So you always have this trade-off, I think, in statistics, and probably in all areas. I mean, it came up in your talk yesterday. So here, our trade-off is, you know, you either have a lot of data, and you can probably do okay with very little computation time, or you have little data. Okay, so if I have an infinite amount of data, I'm happy to use this algorithm, right? Then these things have very little volume. I don't care. If I have a huge amount of data, just go ahead and use it. But, you know, if you don't have a huge amount of data, then you might have to pay the price with computation time. You might have to be okay with using an algorithm which actually needs to do more computations than this algorithm here. That's the price we're going to pay. But at least then we'll understand what are the minimal assumptions needed in order to learn a DAG. And so, okay, so I'm trying to come up with an algorithm which will actually overcome all these 4 points which I said at the beginning. Okay, and I'll explain how it does. Well, okay, one of the points will be still a problem. Okay, so what is the problem of the PC algorithm? Well, I kind of said it, that it puts the emphasis on the wrong place in my point of view, because the goal is not to learn an undirected graph. So you definitely don't want to have a step where you're somehow, just because you're making a mistake when you learn the undirected graph, you're going to make mistakes in ordering in the direction of the edges. So let's just try to get the direction right, put more emphasis on that than on actually learning the correct edges. Okay, so So the goal is to learn the right permutation or the right ordering of the vertices, because then I can, and then learn an undirected graph, and then I can just orient all of the nodes into that direction. Okay, so that's how I'm going to approach the problem, and here's the algorithm. So what does, okay, so it tries to overcome the problem of the PC algorithm. So the PC algorithm tests too many conditional independence relations and just removes edges whenever it finds any conditional independence relation. Right? So that's the problem. So we want to test as few conditional independence relations as possible. And so what is this? So again, I said I'm going to pay the price now hugely with computation time. I'll show you how to overcome this. But for now, I'm going to give myself infinite computation time. I'm allowed to search through all orderings of the nodes. Okay, so I'm taking all orderings of the nodes, and for every ordering, I'm going to construct a DAG. So how do I do this? So I take an ordering. So in this case, I'll take the ordering, for example, well, let's actually do a particular one. Actually, I have it here. Okay. Now I left my thing somewhere. Here. Okay, so let's do a particular example. Okay, so let's say this is my correct DAG, and I have certain conditional independence relations. So for example, 1 independent given 2, 2 independent of 4 given 1, 3, 1 independent of 2 given 4. Okay, how does this algorithm work? So it takes all the orderings. So let me take the correct ordering, 1, 2, 3, 4. What is it going to do? So it's going to test is node I independent of node J given everything which comes before in this ordering. Okay, so for example, is 1 independent of 2 given nothing because there is nothing in front? No, it is not, so I'm going to leave the edge. Okay, then the next thing, is 1 independent of 3 given everything before in the ordering, so given 2? Yes, it is, so I'm going to remove the edge. Then, for example, is 2 independent of 4 given everything before in the ordering, so given 1 and 3? Yes, it is, so I'm going to remove the edge, so on. Okay, so for this ordering, it would learn the correct graph. And if we take a different ordering, I don't know, I'll take anything like 4, 1, 2, 3. For example, you do the same thing. And you'll see that there will be actually 5 edges in here. Because, for example, here, I would be testing is 1 independent of 3 given everything before. Right? Is 1 independent of 3 given 2 and 4? No, it is not, so I would actually have this additional edge inside. So what does this algorithm do? So for every ordering, it's going to construct a DAG. Right? So because I have an ordering, I know in which direction I'm going to put my edges. And then it's just going to choose the permutation with the sparsest DAG. Okay, if there are multiple ones, you just either output all of them or you try output one of them, I don't care. Okay, so that's the algorithm. So you go through all your orderings. Every ordering will give you a DAG. You just output the DAG, which has the least number of edges. And so we can prove that this is consistent under a strictly weaker condition than faithfulness. So in particular, whenever the PC algorithm gives you the correct DAG, this will give you the correct DAG. And it's very easy to come up with an example like this one, where the PC algorithm is going to make a mistake and the SP algorithm is not. And in particular, the assumption needed for the SP algorithm is the minimal assumption needed for any causal inference algorithm to learn a DAG. Right? Because what is the assumption needed? Well, the assumption is that there is no sparser DAG than the correct DAG, which is compatible with the conditional independence relations you have. So if there is a sparser DAG than the correct DAG, then there is really no reason to choose the correct DAG, which has more parameters than the sparser DAG. Okay, so this is a basic, this is an assumption which any algorithm will have to satisfy in order to learn directed graphs. So this is actually the minimal assumption needed for any algorithm to learn, or information theoretic assumption needed for any algorithm to learn the correct DAG. But we paid a huge price in terms of computation time, so we have to still fix that. And I'm just going to give you a little bit of a flavor of how we fixed it, but kind of no results. So there is a working algorithm, but just to give you a little bit of a flavor of how you can fix it. So until now, everything I said about this algorithm has did not make any assumptions on the distribution. So let me just tell you how this algorithm works in the Gaussian setting. Okay, so we're back to the Gaussian setting, where the goal is to figure out the correct ordering and the correct zeros in the Cholesky decomposition. Right? So I want to learn the DAG, and the DAG is encoded in this upper triangular matrix here. I want to learn the correct zeros in there. Now there is a very nice theorem, which actually, okay, so what do we need to do? We need to go through all orderings, right? We need to go through all orderings. For every ordering, we're going to learn a DAG, which means a set of zeros in this Cholesky factor. We're going to choose the one which has the most number of zeros. Okay? Now, the nice thing is that really these zeros in here actually correspond to these conditional independence relations which we are testing in the SP algorithm. Okay, so a 0 in here actually corresponds to the conditional independence relation that, I is independent of j given everything before. So in the Gaussian setting, oops, sorry. So in the oh, I'm done, okay. I won't get to, Shiva will give my second talk on the model. Yeah, I will just finish this, yeah. So in the Gaussian setting, so what this algorithm does is it wants to find, so it wants to go through all orderings and find the Cholesky factor, which has the most zeros. Okay, so that's it. I mean, and so now maybe some of you will know some numerical linear algebra, have heard of this. This is a huge problem in numerical linear algebra. Right? They will have like huge matrices and they want to store them in such a way so that it takes the least number of space. Okay, so they will want to find a good ordering so that all they need to store is a sparse upper triangular matrix that has a huge number of zeros. So this is a problem which has been studied a lot in numerical linear algebra, and that's exactly the kind of problem we're getting to. Just that they don't have noise. We want to find an approximate sparse Cholesky decomposition. But at least you can see how we will get to actually getting some algorithms which run fast and don't really need to go through all orderings. Okay, and this can then be extended to also other settings where you don't really have just Gaussian setting, but you can define something very similar just using ideas from numerical linear algebra to actually get an algorithm with provable guarantees, which can actually also be applied to very large graphs. And okay, so I'll end with, okay, so this took like a huge detour, right? Solving a lot of different math problems before we can actually get back to the biological applications which we're interested in. And I'm not going to show you any results because this is what we're working on at the moment. So we're looking at estimating gene regulatory networks throughout the development of Drosophila, so how they change throughout the development. We're starting with this problem just because there are a lot of data sets on this, and there has been some previous work on actually estimating undirected graphs and how they change over time. So undirected graphs will be much denser, right? There is no causal links in an undirected graph. So what we're really going to do is to get the directed graphs, so the gene regulatory networks and the causal links in there and how they change over time throughout the development in Drosophila. And we're finally going to get back to this learning tissue-specific gene regulatory networks. So this is a collaboration with Lior Pachter at UC Berkeley. So the data sets are from the GTEx project. So what is it? So you have tissue-specific, so you have many different tissues. And every tissue, you have many different samples, and you have various donors. And so in each one of these little Rubik cubes, you can think of as having one vector of size 9,600, because this is how many genes they're sampling. And what we would like to know is, well, we would like to learn gene regulatory networks and how they change throughout different tissues. And maybe also say something about person-specific components in these gene regulatory networks. Okay, and with that, I will not get to the second part of spatial organization of chromosomes, but this is joint work with Shiva, and he's just going to talk about the model as well on Thursday. Okay, and let me just go to the end. How do I go to the end? Is there a no? Okay, I'll skip over everything. And yeah, so here are just some references of these papers. Okay, thank you very much.
So, in the application of these graphical models, right? So your axis is tissues and donors, and different time points. And so, just coming back to this issue of time varying things, how do you get at that?
Yeah, okay, so there you can just talk about Drosophila and development of Drosophila.
Mm-hmm.
Okay, so there are a couple of assumptions you can make. So first of all, you want to be using all information or all data which you have, right? So probably, from one time point to the next, the gene regulatory network does not change as much as when you take from one time point to, I don't know, to a different state, like from larva to pupa, for example. There might probably be a very big jump. But still, you don't want to be throwing away data. So I do not want to learn a gene regulatory network at each one of the time points separately. Okay, you do want to increase your power. And so it's a similar idea as for the algorithm, right? So what you're going to do, you will make some sparsity assumptions. So here, for example, you can assume that the regulatory network changes continuously over time with certain spikes when you actually go from larva to pupa and then to an adult fly. So you can include that in the algorithm. So you're going to merge all the samples with some weights. Okay, so that it continuously varies. And then you will learn a gene regulatory network. And you can actually even learn it. So you'll learn a gene regulatory network from these weights. You will even learn the gene regulatory network as it changes between the time points where you're measuring. Okay, so you'll have like the sparsity assumption where you can glue together all the data sets. You learn a gene regulatory network. And by setting the weights as you want them to be, you will learn all the gene regulatory networks in between. Something similar is what we're using for the tissues. So there, you can think that there are some, you probably don't want to learn one gene regulatory network for each one of the tissues separately, just because you also don't have enough samples to do that and not enough power to do that. So you'll have to do something where you first learn how similar are different tissues to each other. So you can think of building something like a phylogenetic tree for tissues. Okay, and then you have like a distance within a tree of each tissue to another tissue, and you use these differences in order to somehow glue together the data, weigh them in different ways, and then actually be able, and then what you're going to get is somehow you're going to be able to infer gene regulatory networks, not just on the leaves in your different tissues, but actually also in the inner nodes, just by choosing the weights as, to whatever it corresponds to in the inner nodes. So that's how we're approaching these problems. But in order to do that, we first needed to figure out another algorithm. So, yeah.
So you talked with your list of desirable properties.
Oh, yeah, great, yeah.
But two of them were not, so prior distributions and what do you do with loops? Could you touch upon those topics?
Yeah, so prior information, so prior information we can use very, very easily. Okay, so this is a search through permutations. So, you're just not going to search through permutation. If you know that a gene is upstream from another gene, because we're actually first learning the directions and not learning the undirected edges first, this information can very easily be included. So that the problem with the PC algorithm is because it's learning first an undirected graph, it cannot include that kind of information. The second thing about loops, let me go back to the Gaussian setting. Okay, so here's the Gaussian setting. Okay, so in the Gaussian setting, what we're learning is somehow upper triangular matrix, so a Cholesky decomposition, right? This is upper triangular. This corresponds to there not being any loops. But you can easily think of learning a different factorization where, let's say you want to have as few non-zero entries in the lower side, but you are going to allow some. It's just that you're going to learn a different square decomposition, and that's why that will allow you to actually have directed loops as well. Now, we're running this kind of algorithm. We just don't have the proofs yet of when it is actually consistent and when not, because there are identifiability problems that you can have certain non-zero entries. There are different orderings which will give you the same amount of non-zero entries, but will be very different DAGs. But at least this has, just running it, it seems to work pretty well, but we don't have the results yet. But at least there is hope that, this kind of approach you can actually allow for directed cycles.
Do you know something about uniqueness? Or do you know something about uniqueness, or are there multiple permutations that can?
Yeah, so this is okay, so without cycles, you mean?
Yeah, without cycles.
Yeah, okay, so that's something I skipped. Okay, so what you're really learning is an equivalence class of DAGs. I mean, I kind of said that, you know, there is no way of telling apart this and this, right? Just by correlations. I mean, they're going to be correlated and they're going to be correlated here. So really what I'm going to learn is something like this. Okay? And even in the example I had with like mom, you know, grandchild and so on. So this guy here is, you know, these two are dependent given this, independent given this guy, but also in this setting, you know, these two are independent given this guy. So there is no way of, this is called, yeah, so we're really learning an equivalence class, but, you know, there is a difference to this graph, right? So we are going to be able to tell apart this stack from these two. So how would you represent it? Well, this guy you would represent like this, but here you would just represent it by an undirected edge, meaning that we know that there are no such something called V structures, so there are no colliders in here. We know that there are no colliders, but they can be oriented in any direction so that there are no colliders. So really what we're learning is always an equivalence class. And then by additional, if you have some prior knowledge, and of course of what gene comes upstream, then you can very easily somehow you know, figure out which ones of these DAGs.
Within this equivalence class, you can prove uniqueness.
Sorry, within
So
this equivalence?
within the equivalence, so you find a unique such equivalence class. It's not
exactly. Yeah. So that's really, I mean, you cannot even hope to do more using partial correlations. So this is what you're going to get out is a unique equivalence class of DAGs, which you then have to resolve by other methods. There you need experiments. There is just no way of telling the difference between these graphs.
Okay, so if there are no more questions, let's thank Caroline again.
Thank you.
And we have a coffee break now.